So I'm very glad to have an opportunity to talk about my research in this symposium. I would like to thank the Jung Foundation for invitation. Okay, so today I have 4 topics. First, I will introduce structure and biosynthesis of GPI-Anker proteins, and I will report what my research group have accomplished in this research area. And then I will talk about the functional characteristics of the GPI-Anker proteins. And finally, I will deal with the 2 forms of the GPI deficiencies that cause human diseases. So this shows the structure of the GPI-Anker protein. So GPI stands for the glycosyl phosphatidylinosyl, and GPI-Anker proteins are integral membrane proteins expressed on the cell surface. But different from the typical transmembrane proteins, protein part of the GPI-Anker protein does not penetrate to the membrane directly, but instead the glycolipid termed GPI is attached to the carboxy terminus of the protein, and the lipid portion of the GPI anchors the entire protein to the outer leaflet of the plasma membrane. And the lipid part is a phosphatidylinosyl, and the glycan part consists of the core and the side chains. The core of the glycan is conserved among all the GPI-Anker proteins, consisting of the glucosamine, 3 mannoses, and the 2 ethanolamine phosphates. Amino group of the terminal ethanolamine is linked to the carboxy terminus by the amide bond. So it's covalently linked to the protein. The side chains introduce structural variation to the GPI-Anker. The many GPI-Anker proteins have the NS2-galactosamine side chain linked to the first mannose, and this can be further elongated by galactose and sialic acid. In some cases, the 4th mannose is attached as a side chain. In human bodies, more than 160 various proteins use this structure for the membrane anchor. And many of them are cell surface enzymes, such as alkaline phosphatase and 5' nucleotidase. And some are the receptors, such as folate receptors. And some are the adhesion molecules and complement regulators. The prion proteins are the GPI-Anker. Because those functions of those proteins are various, so the GPI biosynthesis is essential for embryogenesis, neurogenesis, immune system, and fertilization. And this shows how GPI-Anker proteins are generated in the cell. The biosynthesis the pointer will be fine. So the biosynthesis occurs in the endoplasmic reticulum ER, and the precursor protein and the GPI glycolipid are synthesized separately in the ER. And the precursor protein has the GPI attachment signal peptide at the carboxy terminus. And the glycolipid is synthesized from PI, phosphatidylinosyl, by stepwise additions of the monosaccharide, fatty acid, and ethanolamine phosphates, and to generate a complete precursor of the glycolipid. And there are 10 reactions are involved to generate the GPI. And the enzyme termed the GPI transaminase recognizes the C-terminal GPI attachment signal sequence of the precursor protein and replaces it with the pre-assembled GPI, making the nascent GPI-Anker proteins. At this stage, the structure of the GPI-Anker is still immature. Then the on the way of the transport of the GPI-Anker protein from the ER through the Golgi to the plasma membrane, the structure maturation reactions occur. I will come back to these maturation steps. On the cell surface, in the plasma membrane, GPI-Anker proteins are mainly associated with the lipid microdomains, also called the rafts. And some of the GPI-Anker proteins can be shed from the cell surface after cleavage of the GPI-Anker moiety by the GPI cleaving enzymes, or GPI-aces. And for the entire pathway, there are 30 genes are involved. And the genes involved in the biosynthesis of GPI and its attachment to proteins are called the PIG genes for the phosphatidylglycan. And the genes involved in the maturation steps are termed P-gap genes for the post-GPI attachment to proteins. So if we look in this process in more detail, and this shows the biosynthesis of GPI and attachment to the protein. For this pathway, 24 genes are involved. And we discovered most of the genes, except the genes in red color. Red ones are discovered by other groups first. And if any one of those PIG genes doesn't work, then the biosynthesis stops there, and the complete GPI-Anker is not attached to the protein. Then the precursor protein, if they don't get anchor, is usually degraded in the cell by the ubiquitination and the proteasome. In some cases, non-anchored protein can be secreted out of the cell as a soluble protein. But the major outcome is intracellular degradation. So the mutation in this pathway causes the GPI-Anker protein deficiency on the cell surface. So this shows the maturation steps. When the anchor is attached to the protein, the structure of the GPI is immature, then the maturation reaction occurs in the ER and in the Golgi. And in the ER, the acyl chain, fatty acyl chain linked to the inositol ring is first removed by the PGAP1 enzyme. Then the ethanolamine phosphate attached to the second mannose is then removed by the PGAP5 enzymes. These the structural changes are critical for the efficient transport of GPI-Anker protein from the ER to the Golgi by the COP2 coated transport vesicles. And in the Golgi, fatty acid remodeling and the side chain modification occur. The fatty acid remodeling is important reactions for the raft association of the GPI-Anker proteins. Usually, the phosphatidylinositol has unsaturated fatty acid like arachidonic acid in the second position. The unsaturated fatty acid is incompatible, is inhibitory to the raft association. So the PGAP3, which is GPI-specific phospholipase A2, removes the unsaturated fatty acid from the position 2. Then the saturated chain, usually the stearic acid, is added back to the second position, making the chain structure compatible with the raft association. So if the fatty acid remodeling doesn't occur, GPI-Anker proteins cannot be associated with a raft. That causes a problem. And then the side chain modification occurs. The PGAP4 is a GPI-specific N-acetylgalactosamine transferase. And then galactosylation is mediated by the B3-GalT4 galactosyl transferase. So the B3-GalT4 is not a GPI-specific galactosyl transferase. It is known as the ganglioside GM1 synthase. So the ganglioside biosynthesis pathway and the GPI-Anker biosynthesis pathway use the same galactose transferase at this step. The sialyl transferase that attaches the sialic acid has not been identified. So we discovered all the PGAP genes. So the biosynthetic pathway of the GPI-Anker proteins are quite well understood. Now I move to the second part, which is the functional features of the GPI-Anker proteins. At least three functional features are known. One is, as I already said, that GPI-Anker proteins are associated with membrane microdomains or rafts. So the membrane microdomains are the dynamic domain rich in sphingolipids, cholesterol, and the signaling proteins such as the SARC protein kinases. So many of the GPI-Anker proteins within the raft are involved in the signal transduction after the ligation of their binding of their ligands. And the second feature of the GPI-Anker protein, the shedding by the GPI aces. There are 4 pairs of GPI-Anker protein substrate, and the GPI aces are known. Those combinations are linked to the specific biological event. For example, the REC is a metaprotease inhibitor, the GPI-Anker protein inhibitor. And when the anchor is cleaved by the phospholipase C activity of the GDE2 enzyme, the REC is removed from the cell surface. Then the suppressed metaprotease become active, and that degrades the notch ligands. Degradation of notch ligands causes the differentiation of the certain type of the neural cell precursor. So this release shedding event is involved in the neurogenesis. Similarly, the UPA receptor is a GPI-Anker protein. If it is removed by the GD3 enzyme-mediated cleavage, then the cell adhesion and migration changes. So those GPI aces are substrate protein specific. So these combinations are linked to the specific biological event. The third feature of the GPI-Anker protein is the transcytosis. In the capillary endothelial cells, GPI-Anker protein called the GPI-HBP1 transports lipoprotein lipase LPL from the subendothelial space to the capillary lumen, thereby allowing the degradation of triglyceride within the chylomicron to fuel tissues with fatty acid. This is an important step of the action of the lipoprotein lipase. If the GPI-HBP1 is deficient, that causes the severe hyperchylomicronemia, like lipoprotein lipase deficiency. So this phenomenon was found by Steve Young, and Dr. Young was received the award in medicine some years ago for this discovery. Now I move to the GPI deficiency. Because the GPI-Anker is important for more than 160 various proteins, if the GPI biosynthesis is completely deficient, it causes the embryonic lethality in the very early stage of the embryogenesis. This was shown in the piglet knockout mouse. There are 2 modes of the incomplete deficiency of the GPI that causes the human diseases. One is the paroxysmal nocturnal hemoglobinuria, PNH. This is an acquired disease of adults caused by the somatic mutation in piglet gene. And the second form is inherited GPI deficiencies. This is inherited disease of kids caused by the hypomorphic germline mutations in pig or the PGAP genes. So I will first explain the PNH. The PNH was first reported by the German physician Paul Strubing in 1882. And in 1932, it was demonstrated that the hemolytic anemia seen in PNH is caused by the defective protection of red cells from complement action. And in the mid-1980s, the molecular basis of the intravascular hemolysis was clarified. Namely, it was found that in the abnormal clonal blood cells, the two complement regulatory proteins, DAF and CD59, are missing. And this makes the affected cell highly sensitive to activated complement. Either the pathway-induced strong activation of the complement or the spontaneous activation of the alternative pathway. And in mid-80s, GPI-Anker proteins were discovered, and DAF and CD59 both are turned to be the GPI-Anker proteins. In 1993, we found that the DAF and CD59 deficiencies are caused by the somatic mutation in PIGA gene. This showed a flow cytometric profile of the DAF and CD59 expression on the normal and the PNH patient blood, red blood cells. All the normal red blood cells express DAF and CD59. Dashed lines are the background. In the patient red cell have two populations. One is a normal population expressing DAF and CD59. The other is the abnormal clonal population deficient in both DAF and CD59. And these large abnormal clonal cells are highly sensitive to complement, and they hemolyzed by their own complement. So in 1993 Cell paper, we reported three things. And we found it is known that DAF and CD59 deficiency is found not only in red cell, but also in the granulocyte and lymphocytes, but only in the blood cells. And we found a mutation, PIGA mutation, in the abnormal lymphocytes, but not in the normal lymphocyte, indicating that the mutation in the somatic mutation is not a germline. And then we found the somatic mutation occurs in the hematopoietic stem cell, because the granulocytes, abnormal lymphocyte have the same mutation. So the mutation must occur in the hematopoietic stem cell. Then we found that PIGA is an X-linked gene. This means one somatic hit causes GPI deficiency. In male stem cell, of course, only one PIGA gene. So the one somatic hit immediately causes the GPI deficiency, making the GPI deficient clone of the stem cell. In the female, there are two PIGA genes, but one of them is inactivated by the X chromosome inactivation. So if the somatic hits to the active allele, then again the one hit causes GPI deficiency. So this is our finding in 1993. So this shows the situation of the PNH. In the patient blood is a mosaic. Have the normal red cell and the big expanded abnormal clonal cell. So this abnormal clonal cell is produced by the clonal expansion of the mutant hematopoietic stem cell. Generates large amount of abnormal cell population. When the complement is activated, the activated complement components are deposited on the red blood cell surface. On the normal red blood cell, DAF inhibits C3 convertase, CD59 prevents membrane attack complex formation, so nothing happens. But on the PNH red cell, there are no regulation, so the activation leads to the generation of the membrane attack complex, and then water comes in through the channel of the membrane complex that leads to the hemolysis and the release of the hemoglobinuria. So this is the mechanism. So the intravascular hemolysis by complement is a major problem of the patient with PNH. But now the intravascular hemolysis is well controlled by the anti-C5 drug. The eculizumab is a humanized anti-C5 monoclonals developed almost 20 years ago. When the eculizumab is bound to the C5 protein, even on the PNH cell surface, C5 convertase is generated. The C5 cannot be cleaved by the C5 convertase. In that way, the eculizumab very efficiently stops the intravascular hemolysis and improves the patient QOL a lot. So the PNH is somehow under control now. Now I move to the inherited GPI deficiency. So the IGD is a relatively newly found disease. In 2005, I was asked by the Dr. Kara Dimitris in London to help clarify the molecular basis of the two consanguineous families with the GPI-Anker protein deficiency, characterized by the seizures and hepatic vein thrombosis. So we obtained the patient cells from London, and my colleague Yoshiko Murakami in the lab tested all the pig genes and found the hypomorphic promoter mutation in pig-M gene. So pig-M encodes the manuscript transferase that attaches the first manus to the GPI. So this is the first cases of the GPI, inherited GPI deficiency. We published it in 2006. And then the second cases are found in Germany. Peter Kravitz in Berlin, Charité, now he is in Bonn, applied whole exome sequencing to the patients with intellectual disability and growth delay, and found PIG-V mutations. The PIG-V encodes another manuscript transferase that attaches second manus to the GPI. And we analyzed the mutations they found and showed that those mutations are hypomorphic mutations, accounting for the partial decrease of the cell surface GPI-Anker proteins. So this is the second report. Since 2010, many IGD patients are discovered, mainly by the whole exome sequencing. And as of February this year, pathogenic mutations are found in the 24 pig and PGAP genes. In total, 800 patients were reported. We contributed to the report of the first cases of the IGD with mutation in those genes are shown in red. So in inherited GPI deficiencies, levels of various GPI-Anker proteins are low because of partial loss of function mutations in pig genes. Or structures of GPI-Anker is abnormal because of narrow or partial loss of function mutation in the PGAP genes involved in the maturation reactions. So the major symptoms are developmental delay, intellectual disability, seizures, and hypotonia. And the other symptoms are visual impairment, hearing loss, the various anomalies in other organs like heart, kidney, and the intestines. So it's very clear the major symptoms are central nervous system related. So the neuronal system is the most sensitive to the partial loss of the GPI-Anker proteins. If mutations are severe, then the abnormality in the other organs appear. So this shows the level of the some of the GPI-Anker proteins on the blood granulocytes from pig A IgD patients. The 5 patients, they have different mutations. And among the various GPI-Anker proteins, CD16B, which is Fc gamma receptor type 3, is the most sensitive to the mutation. And only 5 to 15% of the normal level remained. So this is the best marker to diagnose the IgD. But if you see the DAF, the complement regulator, more than 50% remained. So they still have the ability to regulate complement. So the IgD patient don't get hemolysis. This is very different from PNH. In PNH, almost 100% of DAF are missing. So this is a summary of the symptoms of the 65 cases of PGAP3 IgD. And so those patients are deficient in the fatty acid remodeling. So the association of GPI-Anker proteins to the left is impaired. And almost 100% patients have the global developmental delay, motor delay, cognitive delay, facial dysmorphism, speech delay, and hypotonia. And if mutations are severe, then the other symptoms appear. So again, the central nervous system is the most sensitively affected by the partial loss or the dysfunction of the GPI-Anker proteins. So there is no effective therapy for the IgD at this moment. So because IgD is a monogenic disease causing the deficiency of the specific enzyme, so we thought supplemental gene therapy might be effective for IgD. So for that goal, we established IgD model mice and tested the gene therapy. So we chose the PIG-O gene because the PIG-O IgD is the most prevalent in Japan. So we knocked in one of the patient mutation in PIG-O into mice and generated heterozygous knock-in mice. And by mating heterozygous knock-in mice, we obtained the homozygous knock-in mice in the IgD model. We also generated the heterozygous knockout mouse and mating with the heterozygous knock-in mouse to obtain the compound heterozygous knock-in knockout mouse, which is more severe. So in those knock-in homozygous, knock-in knockout heterozygous, GPI-Anker protein levels are significantly reduced in the blood cell surface. Then we intravenously injected AAV vector bearing the human PIG-O cDNA into the newborn mice. And this AAV vector has the very efficient blood-brain barrier penetration in the C57 Black 6 mice. So the IV injections give the cDNA into the brain. On the left side, it showed the recovery of the GPI-Anker protein 1 month after injection. The recovery is mild, and increase is not statistically significant, but still the clinical efficiency, efficacy is quite strong. On the right side is a knock-in homozygous mouse stopped the growth at 9 to 10 weeks, and the blue line. But the treated mice kept growing like control mice in male. In female, the efficacy was less efficient, but still significant. The knock-in knockout, more severe mice, the growth stopped at earlier time, but efficacy was partial, but still significant. And the model mice showed the tremor and hind limb clasping, a sign of the neurological deficit, and they failed hanging. But the treatment was ameliorate the tremor. The treated mice have weaker tremor scores. The appearance of trembling is slower, and hind limb clasping disappeared. And treated mice stayed in hanging a much longer time. So the single injection, intravenous injection of the AAV is quite efficient for the amelioration of the many neurological symptoms and the growth. So in summary, there's a PIGO mutation knocking mice reproduced symptoms of IGD, such as growth delay and neurological deficits. AAV-based gene therapy of newborn model mice was effective. So the gene therapy is promising treatment option for IGD. My colleague Yoshiko Murakami works hard now to realize the supplemental gene therapy for the human patients. So I will stop there, and I would like to thank my colleague Yoshiko Murakami, Junji Takeda, and lab members. I also would like to thank the Japanese collaborators and the international collaborators, particularly to the Dr. Karan Dimitris in London and Dr. Peter Kravitz, now in Bonn. And finally, vielen Dank für die Einladung der Jung-Stiftung. Thank you very much for your kind attention.